Think forward. Think Research Channel.
This is the Dean's Distinguished Lecture Series. And if it's the Dean's Distinguished Lecture Series, we owe you a distinguished colleague, a distinguished scientist, a distinguished lecturer, and that is certainly the case today. Our speaker today is one of our own, Dr. Gary Van Zant, and I want to provide just a brief introduction, which probably won't do him justice, but I think it's always fun to step back and see who we are and where we came from. Dr. Van Zant got his bachelor's and master's degree at the University of Nebraska and his doctorate in cell biology and hematology at New York University. He then rose through the ranks in various institutions. Before coming here, he was at the University of Michigan. And Dr. Van Zant joined us in 1995 as professor of medicine. Now, he does a lot of things, and I don't want to overlook anything, so I'm going to read. He's professor and clinical director of the Stem Cell Processing Lab in Markey and in the Division of Hematology and Oncology. He's the director of the Cord Blood Bank here at the Medical Center. And he also has secondary but important appointments in pathology and physiology. Gary's lab, as you probably know, focuses on age-related changes in hematopoietic stem cell function. And indeed, his lecture today is entitled Genetics and the Effect of Aging on Stem Cell Regulation. Dr. Van Zant, please. We like to reward Gary in anticipation. So, in fact, that's what I'd like to do. I'd like to offer you a token of our appreciation, not just for doing this lecture, but for being a terrific faculty member.
Thank you very much. Thank you. Pleasure to be here, and I want to thank Dr. Bakke and Dr. Perman for inviting me to present this in this prestigious lecture series. I think that I'll go right to the objectives today. I want to present two major foci. One is to present a genetic approach that we've used to identify regulators of adult stem cells. And I should discriminate here between adult and embryonic stem cells. We work on adult stem cells in the body, particularly those in the bone marrow that fuel the production, the continuous production of blood cells throughout our lifespan. Now the second part of the, and this is going to be a genetic study. Now the second part of the talk is going to show how this can segue into a translational approach to aiding in, hopefully, in clinical treatments and particularly in the treatment of cancer. So we have basic research that has led us through several years to a translational point in the story that I'm going to tell you today. So the story begins with a tale of two different mouse strains. Now mouse strains are nice because they're all inbred and we don't have to worry about genetic variation among animals of the same strain. And so for genetic studies, they're ideal. And these are the two that we chose. These are our poster children here. And what do they differ? How do they differ and why are we interested in them? The strain on the right, DBA2, has a lot more stem cells when young compared to the black 6 strain. The stem cells turn over at a much more rapid pace in the DBA strain than the black 6 strain. In old age, perhaps not surprisingly, the DBA strain has fewer stem cells, and the black 6 strain has more. And lastly, the DBA strain in relationship to the black 6 strain is short-lived. And we believe that these 4 different phenotypes are closely related, and we're attempting to show that. But today, I'm going to talk about only one of these phenotypes. I'll show you in a minute. Now, the story begins many years ago, actually, when we made cellular chimeras between these 2 strains of mice. And what we did was harvest 8-cell embryos from these 2 strains. We aggregated the embryos, so we mixed the cells of the embryo. We transplanted those cells into pseudopregnant females who carried them to term and gave birth to these cellular chimeras. These cellular chimeras consist of a mixture of cells, not genes, but cells. So they're cellular chimeras, and they consist of a mixture of cells in all the different organs of the body. And the one we were, of course, interested in was the hematopoietic system. And so we analyzed the chimerism in the blood cells of these animals during their lifetime. So to run quickly through the hematopoietic hierarchy, it begins with stem cells. And in these chimeric animals, we had both DBA and black 6. This thicker arrow here is to allude to the fact that they turn over at a more rapid rate than do those of the black 6 origin. The multipotent progenitors are also a mixture of both, and so on down through the hierarchy until we get to the mature blood cells, which also consist of a mixture of DBA and black 6 cells. And our real interest then was the chimerism over time in these animals should reflect the relative contributions of the 2 stem cell populations during their lifetime. And we expected there to be some differences, but we were very, very surprised to see that after an initial period of about a year, when the chimerism in these animals was roughly stable and roughly 50-50, things began to change. And the contribution from the DBA stem cells began to drop out of the picture until after 30 months or so of the lifespan of these animals, the DBA contributions to the hematopoietic system completely stopped. Now, the animals didn't die. They continued to live just fine, but their blood system and lymphoid system was completely derived from black 6 stem cells. So this shows a differential aging and a differential stem cell number in these animals that is strain specific. And so what did the experiment really tell us? Well, we learned that when the stem cells coexisted in the same environment, they displayed strain-specific functional differences. They were exposed to the same microenvironment. They were exposed to the same proteins in the serum of the animal, but they maintained strain-specific differences. And therefore, at least some of these traits that resulted in the loss of DBA stem cells had to be cell autonomous. They had to reside within the stem cell itself and not be extrinsically regulated. And also told us that the genetic differences between these strains were important. So we embarked on what's called a forward genetic approach. We had already identified that in fact there were strain-specific phenotypes. What we wanted to know was over here at the right-hand side of the slide. What are the genes underlying these intrinsic effects of stem cells, their aging, their numbers, their turnover, etc. And there are multiple steps in this pathway as we envisioned it. And I will go over these as I progress through the talk. So we'd identified strain-specific phenotypes. We wanted to concentrate on one of these. And the one that I've chosen to talk about in this talk is this one. The fact that DBA mice have more stem cells when young, and that the black 6 strain has many fewer. And this is the trait that we went after. And most of the results of this study were published in February in Nature Genetics with Ing Liang as first author from our lab. Okay, so we knew, and this slide shows, that there is a natural variation in the number of hematopoietic stem cell number between these two strains when young. And depending on the assay used, the difference is somewhere on the order of 3 to 7-fold. This is a large variation. These animals are not mutants. These are normal, commonly used strains, inbred strains of mice, and yet they have this large difference in hematopoietic stem cell numbers. To proceed then with the genetic analysis of the genes that were responsible for this difference, we used a genetic mapping panel, and I'm not going to go, I don't have time to go into the details of this mapping panel, but the mapping panel is called recombinant inbred strains of mice. And these are derived originally from the black 6 and the DBA strains. And they can be used to relate the phenotype to the genotype through linkage analysis, which is a common genetic tool. And this summarizes what this analysis, which I have more or less trivialized here, boils down to. This is a linkage map, if you will, linking genetic markers in these recombinant inbred mapping panel to the phenotype that each of those strains exhibits. And there are about 30 of those strains. And this is simply a regression analysis between the phenotype and the genotype. And these peaks here indicate areas on the genome where the two coincide or are linked most tightly. Now, the first thing to notice is that there are multiple loci throughout the genome. By the way, these are the chromosome numbers across the top here. Mice have 19 autosomes. Multiple loci contribute to this complex phenotype. That's not surprising. This is a quantitative trait, and so you would expect more than one gene to contribute. This isn't a Mendelian trait, so it's not as simple to study. This is a statistical threshold that above which the linkage is statistically important. And you can see that 3 of the loci reach or exceed that level. One on chromosome 3, this is a marker that's a genetic marker that's used in the regression analysis. One on chromosome 3, one on chromosome 5, and one on chromosome 18. The talk that I'm giving today concentrates on the discovery of the gene underlying the locus on chromosome 3. So we've identified a quantitative trait locus that contributes to the trait we're interested in. And in our paradigm, we thought that the best next step would be to create a congenic mouse. Well, what is a congenic mouse? Well, it's pretty simple. What you do is backcross, using the technique of backcrossing. You move a piece of the genome, in our case, encompassing the quantitative trait locus that we'd already identified, and we move it from a black 6 mouse here onto a DBA background. And we do the converse, or the reciprocal. We move a small piece of the chromosome from DBA onto black 6 background. And so chromosome 3 in the congenic animals consists of all genes that are black 6-like, except this one segment, which came from DBA. And the opposite is true down here. And so these are called congenic mice. The designation is, this means that the black 6 is the background strain, and the introgressed portion of the genome that's significant is from D2, and it's this small piece of chromosome 3. Now, I should add that this small piece constitutes less than 1% of the entire genome of the animal. So it's a very small piece. All the other chromosomes in the congenic animal remain of the background strain. And so you ask, what is the effect of this small number of genes that you've moved from one genome to the other? It's still a large piece. It consists of several hundred genes, but at least you start to narrow down and get at the genes that are important here. And so this is a blow-up of the map, then, of the two congenic strains. Here are the two congenic strains. This is on a DBA background indicated by this tan color. The black is the piece that we move from black 6, and the converse, this tan color is the piece that we move from DBA onto a black 6 background. And if we blow that up a little bit, here's the consensus congenic interval. It extends from 19 to 33 centimorgans, so it's not that big. And here is the marker of highest linkage, and it sits right in the middle of the consensus congenic interval. So if we were ever going to be able to identify genes in that congenic interval, we were well situated to do so with this pair of congenic animals. Well, anytime that you make a congenic animal, the important question is, does the phenotype follow the genotype? You can move genes, but unless they have a phenotypic effect, they're useless in mapping or further genetic studies. And this slide simply shows that indeed the phenotype moves with the genotype. And this shows our two congenic strains here. This shows that if you move a DBA piece onto a black 6 background, the number of stem cells increases, and conversely, if you move the black 6 piece onto a DBA background, the number diminishes greatly. So does the phenotype follow the genotype? And it's a resounding yes. Well, what are the possible mechanisms affecting stem cell numbers? Two come to mind immediately. One is that stem cells in one of the strains could replicate at a faster rate. They could self-renew more effectively. And another possible physiologic mechanism would be apoptosis. Perhaps one loses stem cells at a faster rate than the other one and diminishes their stem cell number. So what Eng did, was first look at replication. And you can see that, as I've already said, black 6 has a lower replication rate than does DBA. And if you move the appropriate intervals, it changes the replication rate dramatically. The presence of DBA alleles increases the replication rate dramatically, and if the black 6 alleles diminish it considerably. Let's look at apoptosis. So, sure enough, black 6 has a higher natural apoptotic rate than does DBA inherently. This could contribute to lower numbers of stem cells. And if you move the DBA alleles onto the black 6 background, you diminish the apoptotic rate significantly. We were in for a little surprise when we moved the black 6 intervals onto a DBA background. Instead of increasing the apoptotic rate like we expected, we got a further and significant decrease in apoptosis. We don't understand that exactly. It's probably due to modifier genes in the DBA background. But whatever the reason, it's clear that apoptosis, in addition to self-replication, plays an important role in this phenotype of the stem cell population size. So the next question was, what are the genes? So we had these nice congenic mouse models. We knew that they had effects on stem cells. We knew that the physiologic mechanisms that we suspected were affected by these genes, but what were the genes? And so the way we chose to do that was by looking at the differential expression within stem cells of the two different background strains. And we did that with microarrays. And it was hoped then that we would identify candidate genes and that one of those candidate genes would be the quantitative trait gene that we were looking for, the pot of gold at the end of the rainbow, if you will. And so what Eng did was sort stem cells in a prodigious task that took a lot of mice in order to get these rare stem cell populations, extracted the RNA, labeled it, hybridized to Affymetrix chips, did the expression analysis, and with the help of the statistical department here, we analyzed the expression differences. And what Eng did was analyze 30 to 40 mice from each of these 4 strains, the 2 background strains, black 6 and DBA, and the 2 congenic strains. So she did 2 comparisons. We looked at this congenic strain versus its background strain, and this congenic strain versus its background strain. And in stem cells of these 2, 96 genes were differentially expressed by microarray. And in this comparison, 84 genes were differentially expressed. 17 of those genes were common to both groups. So that narrowed it down to 17 genes. 9 of these genes, of the 17, were regulated in opposite directions. That is, the B alleles went up in one strain and the D alleles went down in the other strain, or vice versa, or their expression went up or down in the 2 strains. And that's what we were looking for, opposite effects. And only one of those 9 genes actually occurred within the consensus congenic interval. And that gene was latexin. And so the question was, what is latexin? It sounds like a synthetic rubber product, but obviously it's not. It was first identified in neurons of the lateral neocortex of the brain, and hence the name Latexin, latexin. It's also detected in a wide range of other tissues, in rats, humans, and mice. And I won't go through the list, but you can see that it's pretty wide-ranging. Latexin is a small molecule, about 29 kilodaltons, only 222 amino acids. its only known function is it's the only known endogenous carboxypeptidase A inhibitor in mammals. That doesn't sound like something that would have a lot to do with stem cells, but that's the only function that was known. And it plays a clear role in the regional specification of brain development. Well, anytime that you get microarray results, you need to verify them by other methods. And the usual method is to use quantitative RT-PCR. And that's what Ng did. She looked at the expression in a wide variety of different cells in the hematopoietic system, ranging from peripheral blood to whole bone marrow to progenitor cells, or what we call Lin-minus cells, and then LSK cells, which is a sorting nomenclature for a population that's highly enriched for stem cells. And the first thing you'll notice is that as one enriches for stem cells, the overall expression of Latexin goes up. So it's more or less concentrated in the stem cell population. But fortuitously for us, it's also expressed in downstream cells, which allowed us to look at proteins later. It's impossible to get enough stem cells to look at proteins, but we could look at them in whole bone marrow cells and Lin-minus cells, for example. The other thing to notice is that you get these characteristic differences between the background strain animals and the reciprocal congenic strains. But it's opposite of what you might expect. Notice that the expression of Latexin in black 6 stem cells is higher than in DBA. So Latexin turns out to be a negative regulator of stem cell numbers. And here we simply compare the two congenic strains. Again, this is the background strain level, depending on whether it's stem cell number or full difference in Latexin expression. And you can see that the introgression of these DBA alleles onto a black 6 background increase the number of stem cells I've already showed you, but it also decreases the expression of Latexin. And the converse is true for the other congenic. Low stem cell numbers, high Latexin expression. So what happens is this also carried through to the level of the protein. We've talked about transcription, but we haven't talked about translation. And this slide simply shows by Western blot that in bone marrow and progenitor cells, the black 6 animals express higher levels of Latexin protein than do DBA. The next thing that we wanted to do was overexpress Latexin. The prediction would be if we overexpress Latexin, we would decrease stem cells. And Ng devised retroviral vectors that carried the Latexin gene in it, and we infected hematopoietic stem cells and asked then, what effect does it have on stem cell numbers? And the answer is that in both DBA and in the congenic strain that carries the DBA alleles, overexpression of Latexin diminishes stem cell numbers, just like we predicted. So let's summarize the relationship between Latexin stem cell number and some of these functions I've talked about. And you'll see that in either, when you compare the B6 and the D2 allele, you get opposite effects. When you have Latexin expression, you have differential effects on stem cell number. Self-renewal is reduced, at least in the black 6. Cell cycling is reciprocally changed depending on the allele, as is apoptosis. So there are two major questions now. What are the genetic differences between the black 6 and the D2 latexin alleles that result in differential expression? And what are the molecular mechanisms through which latexin regulates stem cell function? We've already talked about some of the physiologic changes, cell replication and apoptosis, but what are the molecular mechanisms that are important? So let me first describe a little bit about the latexin gene. The latexin gene is unusual in that it is transcribed from the minus strand of DNA, and what's more surprising, it sits in the intron of another gene. And this other gene is GFM1. This is a elongation factor that results in the elongation of translated proteins in mitochondria. It is not differentially expressed, we looked, in any of these congenics or background strains that we're talking about. But the latexin gene sits in this intron and is transcribed on the minus strand. I want you to concentrate now on the SNPs, or the single nucleotide polymorphisms, that are different between the B6 and D2 alleles. And there are, as you can see, some 19 or 20 SNPs in this region that's indicated here. We were interested in these up here because these are near the transcriptional start site for latexin. And you can see that there are 3 clusters of these SNPs. This one, and this one. Now these 2 tracks here, SBIR is an algorithm, a computer algorithm that mines genomic data, and it predicts regulatory regions in DNA. And so when we use the SBIR program, you can see that these are putative regulatory regions of the latexin gene control. And this track, called traffic, predicts the binding of transcription factors. And it predicts transcription factors would be bound here and in this region here. And you'll notice that these clusters of SNPs coincide with these regulatory elements here and potential transcription binding sites in in purple here. I've talked about these other 2 points about latexin, but this is the one that takes us into the translational realm. So latexin has about 30% sequence similarity, but much greater structural homology, over 80%, with a human tumor suppressor gene that's called TIG1, or tazarotene-induced gene 1. And so the hypothesis that we're now working on is that latexin acts as a tumor suppressor, and it plays a role in tumorigenesis. And we've begun to look at this. And so the first thing that we did was look at human cells, normal human cells, and we looked at cells from patients with AML and lymphoma and leukemia cell lines. And you can see immediately that the expression of latexin is much higher in normal cells. These are CD34 stem cell enriched populations of cells. They're much higher in AML or lymphoma, and in leukemia cell lines sometimes they're even absent. And so this is consistent with latexin being a tumor suppressor gene, because you would expect it to be downregulated in these tumor cells and tumor cell lines, and this fits. So cancer usually arises from, and this is a simplistic cartoon, but it arises from interactions between 2 different types of genes, tumor suppressor genes and oncogenes. And methylation has been shown recently, epigenetics and methylation, has recently been shown to be important in the regulation of both of these families of genes. In oncogenes, what you have is the demethylation of these CPGs, or these CG dinucleotides, that usually lie in a CPG island that precedes the transcriptional start site of a gene for transcription. This hypomethylation, in the case of the oncogene, leads to transcriptional activation and oncogene expression. Now, in the tumor suppressor, what you usually have is hypermethylation. So these CPGs are hypermethylated, indicated by these closed circles, and you get a methylation binding domain that sets up on these methylated CPGs that prevents the binding of transcription factors and prevents the initial events leading to the transcription of a gene, of a tumor suppressor gene. And so you have transcriptional repression and loss of tumor suppressor expression. And both of these work in concert to lead to cancer. So what Ing did next then was look at these CPGs in the promoter region of Latexin, and there are 15 of them here that she identified. Here's the transcriptional start site for Latexin, and we wondered if we demethylated these CPGs, whether we could reinitiate the transcription of Latexin in some of these cell lines that had either no Latexin transcription normally or very low transcription levels. And you can see in these colored bars here, these purple bars, that in every case, demethylation resulted in the upregulation of Latexin expression. So it looks like Latexin can be regulated by methylation and perhaps other epigenetic mechanisms. So in a heroic effort, Ing also did what is the gold standard for determining methylation, and that's bisulfite sequencing. And each of these circles represents one of these CPGs that I showed you in the diagram of the Latexin gene. And so there are 15 of these. And she looked at each one of the 15 and determined whether it was methylated or not. And these are CD34 positive cells from young donor. And she looked at 8 different clones, and not a single CPG was methylated in this. This is also an old donor. We thought age might be important here, but again, there were no methylated CPGs, and as you might expect, the expression of Latexin was high in both cases. She then looked at AML, acute myelogenous leukemia, and looked at 2 patients. And she highly purified the leukemic stem cells from these 2 patients by using CD34 and also CD38 negative. And this is a very rare population of cells. And she again looked at the methylation status for each of the CPGs. And in these 2 patients, what she found was that some of the CPGs were methylated, and they fell into particular areas. Methylation, CPG site number 4, 10, and 11 were methylated in this patient. 8, 10, and 13 were methylated in this patient. You notice that 10 is the same in both patients. We've only looked at 2 patients in this much detail, but we're looking at more, obviously. And what's the outcome in terms of Latexin expression? It's very low, as you might expect, in both of these AML patients, and specifically in these very rare cancer stem cells. So, why those sites? What's important about those sites in the different patients that might be important? Well, doing data mining, we can predict what different transcription factors bind to regions where these CPGs occur, and where they're methylated in these Latexin, in the Latexin gene. And site number 4 binds a transcription factor called HSF2. 8 binds a transcription factor TTK, and you can read the rest of them. 11 is obviously a very important one, MIB, and CREL at 13, and NF-kappa B also at 13. So we have some candidate transcription factors whose binding to the promoter region of Latexin may be important in its regulation. And this is what we're now pursuing. So the conclusions then are Latexin expression is regulated by polymorphisms in the regulatory regions. We know that there are plenty of SNPs, and some of them are probably important. And we also know that epigenetic regulation occurs in Latexin, and that these two are probably related. So what are our ongoing and future studies here? We want to investigate, we've only looked at the hematopoietic system. We want to investigate the Latexin expression and dysregulation, perhaps, in other types of cancers. And lung cancer stem cells, for example, or cell lines, show dramatic Latexin dysregulation. We know that. That's one candidate. The other thing we're pursuing right now, we're very interested, so we've patented Latexin for the uses in the regulation of stem cells and the and treatment of cancer. We're obviously interested in identifying small molecules that can affect Latexin activity, and we want to develop therapeutic strategies that are clinically applicable. And so, in conjunction with a consortium that UK has set up with the Genome Research Institute up at University of Cincinnati, we are screening a very, very large library of small molecules for ones that have medicinal use in regulating Latexin. We're making a Latexin knockout mouse because obviously if we knock out Latexin, we want to know what effect it has on tumorigenesis in these animals and in different organs. And so we're making a conditional knockout of Latexin so that we can knock Latexin out in specific organs, specific tissues, after various treatments, and at any time that we want, so that we can study the timing and the tissue specificity of Latexin action in cancer formation. And we're taking advantage of some very new advances in proteomics, to high throughput methods, to identify the binding partners for Latexin intracellularly, so that we can uncover the molecular and mechanistic pathways that regulate Latexin action. And those studies are all ongoing. All of these studies are ongoing. We're about 9 months away from having our Latexin knockout mouse. So here's Ing Liang, who was a graduate student in the lab, is now a postdoc in the lab, a proud product of the physiology department, and she did the vast majority of the work here. And to her, I'm very grateful. I'm also grateful to the other people in the lab who have contributed, graduate students, technicians, and 2 former postdocs who were involved in the very early stages of this project and have gone on to their own independent careers. One of whom, Art McGeiger, is a candidate for a faculty position here, and we're hoping to recruit him. And lastly, there's a lot of thanks to be spread around to those who helped us get this work done. And obviously the most important were grants from the NIH. These animal studies are extremely expensive. These genetic studies are extremely expensive. And we've been lucky to be well funded over the years to be able to do this. I've enjoyed the continuous and strong support of the Department of Internal Medicine and the Division of Hematology Oncology over the years, the Markey Cancer Center. I'm indebted to the Department of Physiology, where I have my joint appointment, and for the marvelous group of graduate students who have contributed to this and other projects. I'm grateful to the Sanders Brown Center of Aging for support for a number of these other studies, and the Office of the Vice President of Research, especially with regard to the small molecule screening approach and the consortium with the Genome Research Institutes. So thank you for your interest and attendance at this talk. I'd be happy to take any questions. Thank you very much.